Okay, so this first part should seem like old news to you. We are taking our test. This is 5th hour. So we're taking our test Wednesday. We have 61 questions. It'll be in Illuminate Ed, the same as we've been doing all along, live on the Zoom. They're all multiple choice, no short answers. This PowerPoint, like every day we've been doing Q&A with different chapters. It was part of the daily agenda. I just made a separate file and I called it, you can see, I called it the Q&A. So this is in Google Classroom. Each color is the next chapter. So if you wanted to review any of the Q&A we've already done, you can go back and open this up. We're on chapter 8, which is the blue. So the questions I've seen come up is, how do pigments work? So basically they absorb certain wavelengths of light and they reflect others, right? So white light is made up of all the wavelengths, Roy G. Biv. And then like this blue rock is reflecting blue, so it's absorbing green and red. And this is probably more relevant to plants and chlorophyll. And the chlorophyll will absorb the red and blue, but it reflects the green. So it's getting energy from the red and blue lights, and it's giving those energy to the electrons. And the electrons are taking that into the electron transport chain. So then there was questions about the electron transport chain. And they're taking the energy, they're splitting water, they're releasing oxygen, and the electrons are going to go to make either ATP right here or make NADPH right here. Those two things are going to be used in the Calvin cycle. So the Calvin cycle, you know what, this is a little out of order. So let me move this one down. Oh, well, not that far. Okay. So then the three things that are happening during photosynthesis, basically the 3 stages, you can look in your book, page 223 if you want, 226. You have the light absorption phase, which is the one I just showed you, the electron transport chain, which I showed you, and then the Calvin cycle, which is going to use CO2, ATP, and NADPH from the first set of reactions to make glucose. So you don't need to know all of the steps that we learned, like we learned them before. You basically just need to know what goes in and what goes out. You also need to know where these things are happening, right? So light reactions, this is all happening in the thylakoid, and the Calvin cycle in the stroma. That was another question that you guys had already answered. The light independent relies on light. Notice my my speaker notes, that's why I have it open. And they come first. These are things you might see on the test. Light independent reactions may refer to Calvin cycle, carbon fixation, I actually can't see, dark reactions, all different names meaning the same thing. So here's the chart a lot of you will want to add on. So I'm going to give you a minute to add on to your chart. Many of you just put like the big molecule, glucose, CO2, or water. Over here I have the page number. If you're like not sure about the information, you can go to the page number. You want to know the location in the cell. You want to know what's being made, what's being produced. These things that I highlighted in blue, pay special attention to. So that kind of directs you a little bit more as far as the test goes, what you can expect to be asked of you. So I'm just giving you a couple minutes to fill this chart in. We did a lot of the review really well. Some of the things we just needed some more detail on. And I noticed the tables are where we definitely could use a little more detail. So in Google Classroom, this file is the most recent thing I put up. And it was like class-driven Q&A. So these are the things you guys have asked me over the past week. Anybody need more time on this page? Okay, I'm gonna keep going. You have that slide if you need it. 36 was a number many of us missed. So make sure when you're doing your numbers of ATP production, aerobic respiration makes 36 ATP. I got a lot of wrong answers in this one point. Most of us had electron transport as 32 and fermentation as 2. So good job with that. You can see over here, glycolysis makes 2, Krebs cycle makes 2, and 32 from electron transport makes 36 together. 32 plus 2 plus 4. You want to know the majority of ATP is made in the electron transport chain? And if you pay attention to ATP from fermentation, 36 from cellular respiration, that means it's 18 times more efficient, right? More effective. Those are the kind of things you would expect to see on the test. Most of us have this, but this was a couple people had asked about who does what. So alcohol fermentation takes place in yeast, and lactic acid fermentation takes place in animals. And you could say for both of them, some bacteria. So associate lactic acid with muscles of animals, that's that burn you feel when you work out, and alcohol fermentation with yeast. Think of bread dough, it smells a little. So that was all of the questions I got. Actually, I had a last minute question come in. Somebody had asked about the dark reactions. And basically, like I said, you don't need to know all of the steps. Just know what goes in and what comes out. Carbon dioxide's going in, ATP and NADPH going in, glucose comes out. Okay, so those are all the Q&As that students have given me. Are there any from any of the chapters you want me to go over? You can throw it in the chat or you can shout it out. Any others that you want me to go over? I'll show you a couple things that you guys have available to you in Google Classroom. Sorry, I hate when I do that. So I'm going to go to 5th hour here if it goes. This work? Okay, so we just did on the class-driven Q&A. Oh, I'm sorry, I didn't hear. I see 3 in chapter 6 and go over the importance of gap 1 synthesis gap 2. Yep, I'll do those. We're going to do a Kahoot today. So that's linked in with today's topic. And we're also going to do a Quizlet live if you want for the fun of it. That's what last hour chose to do. So I'm just showing you a couple of things before I get into those last questions. Another thing that you may have forgotten about over here under class resources. There you go. Just like that help. I keep saying 1:30, but you can go by yourself if you want. That's fine. Okay, I'll be done shortly. Under class resources, you have, not class resources, I want helpful links. Sorry, you guys, for that. You have this link here, Snook Bio Reteach Videos. So if there's a topic that right now you're just unfamiliar with and you want to go back and study something, here's a place where all these videos are put into one place. I need to add osmosis because we really struggled with osmosis. So if you clicked on these slides, they'll take you to a page and then the video will launch. So that's a review thing you can use for a specific topic if you're struggling. The other thing I wanted to show you is, let's go back into classwork and you're like, I want to review glycolysis, but I don't remember when we studied it. So I don't know if you know this little trick. The F3 key. Now on my keyboard, I have to hit function and F3, but that's because my function keys have more than one job. So if your F3 key is all by itself, you can just hit F3. But if you hit that, you're going to get a search tool up here. Do you see up here? So I can write glycolysis because I want to study it, but I don't remember when we did it. So it'll bring me to that area. So here I can see week 15, we studied glycolysis. So that'll help me jump right away to the things I want to study. Or I want to study organic molecules. it jumped right to week 5. And it shows me where I study it, highlights it. So F3 key can be really helpful in a quick search. Otherwise, we have 19 weeks worth of stuff to scroll through. So you're like, I don't remember where we talked about the Kelvin cycle. So type that in. I lost it. I must not have Kelvin cycle as a keyword. Light reactions. There you go. Week 12. So those are some things you can use to help you out. Now, the last 2 questions. I'm pulling this back up. So I have a question about, okay, yep. So if you have Chromebooks, you hit Control F. Thank you. So in chapter 9, there was a question about G1, S, and G2. What's the importance of G1 or gap, synthesis, and gap 2? So in the gap phase, this is an increase in size. I think I asked you on the test, where does it do the majority of its growing? The majority of the growth happens during G1. When is DNA replicated? S. S stands for synthesis, and that's what's being synthesized. G2 is just replication of spindle fibers and organelles. I'm not sure G2, I asked you a question on the test. G1 and S definitely have questions on the test. And then number 3 in chapter 6. Identify the role of enzymes in chemical reactions using lactose and lactose. So lactose, OSE endings are enzyme sugars, right? ASE endings are enzymes. So lactase is the enzyme that breaks down lactose. So how does it do that? How do any enzymes work, right? They lower activation energy, or number 3 in chapter 1. So let's go back up. Oh, this one. Okay, so the control group is the setup that does not have the independent variable. I'm just going to write no independent variable. Experimental group is the setup that has, so I'm going to do with independent variable, has the independent variable. Independent variable is the thing the scientist changes. And I'll give you examples here in a second. Dependent variable is the thing the scientist measures as a result of the independent variable. Hypothesis is an educated guess. So an example, I'm going to use the effect of different colors of light on plant growth. So my independent variable are colors of light. I'm going to measure the height of my plant. So these are going to be plants with red, green, or blue light. The control will be plants with natural light. My hypothesis is if plants are subjected to, they absorb red and blue, that seems like that would make them grow better. Red or blue light, then they will grow taller than plants grown in green or under. So I personally manipulate the independent variable. I measure the effect of the independent variable. I have different setups, those that contain the independent variable, those that don't. And then I have an idea of how I think it's going to turn out already. So you'll be given some situations, experimental situations, and you're asked to identify from this, what is the independent variable? What would be the dependent variable? What is the hypothesis the scientist is trying to test? That sort of thing. So the hypothesis usually is stated if independent variable, then dependent variable. So if grown under color, then grows better. Dependent variable. Okay. Any others you would like me to go over from any of the chapters? It was asked if they're all multiple choice. They are all multiple choice, 61 multiple choice questions. Those of you who are fast, probably going to get it done in a half hour. Those of you who take a little bit more time, probably an hour. Any other questions on this?